Before that time.
The physics behind, for example,
another question
the actual change in
under
mass.
the ultimate question gets to evaluate profit.
Over the course of 7 years, the book was put together.
Welcome to the Scholars Chair. Tonight, a conversation with Dr. Sridhar Hannenhalli, Professor of Cellular Biology and Molecular Genetics from the University of Maryland. And here is a clip of introduction.
My name is Sridhar Hannenhalli. I am a professor in cell biology and molecular genetics, and I'm also affiliated with computer science. The broad million-dollar question in the field of biomedicine is, how does individual's genome determine the person's susceptibility to different diseases? Everybody in the field is trying to understand this relationship. It goes through many, many layers of control. The first layer of control is, how do specific genes get turned on to make these messenger RNAs? As a computational biologist, I am interested in understanding how different genes and proteins in a cell go on and off depending on what part of the body they are in and what state, what healthy or disease state we are in. Ultimately, everything hinges on that process as to what makes it manifest itself into a phenotype or a disease or what have you. So we are looking for needle in a haystack. And when we do that, we need lots and lots of data to go through to make sure the statistical inference we are making are robust. Without the huge amount of data that is being generated and without the power to access and compute, process all this data, we could not even be asking the questions we are daring to ask today. As a scientist, I'm driven by basic curiosity. It still boggles my mind how different cells of the body, which have identical genomes, do completely different things. My heart is totally different from my skin, for example. How does that happen? So that's what keeps me coming up to work every day.
For the next 30 minutes, our interview with Dr. Hannenhalli concerning the new development of genetics, right here in the Scholars Chair.
Let the conditions of inequality that we suffered under, when we are the oppressor, the dominant class, let's not impose that injustice on the others.
Anybody think that you could have a just system without giving women their rights?
Everyone has an opinion, and we sit around the marketplace and talk about opinion, but what is true?
With the shareholder, their goal is similar to the business, to maximize profit.
That belief becomes a context for a development of knowledge.
Say physics is the DNA of technology because the rules for how you build new technology starts in physics.
Because Quran challenges the people. It's not only the people of the book, it challenges Muslims.
We say secular, they hear godless.
Right.
What was intended?
Watch the Scholars Chair every Monday night. And here is your host. Welcome to the Scholars Chair, Dr. Hannenhalli. This is a first for us, and we're glad to have you here. My first question to you is, what is the genome?
The genome, it's the blueprint of life. All living things, all cells have genome in them. It could be DNA, it could be RNA. And it's basically tells the cell what proteins to produce and how to live in the environment they live in. And when the cells make up a body like ours, then much more has to happen than individual cells doing their business. They have to coordinate as a tissue, as an organ, as a whole living thing. So ultimately, all this information is encoded in this thing called genome, which lives inside the cell.
Now, it reminds me of the Genome Project that Francis Collins and Greg Venter were pioneering. What do they offer in your study of the genome?
So it has been long recognized that to understand the variety of diseases we face, genetic diseases, to understand them and to treat them eventually, we need to know what is causing them. And because everything that happens to us in terms of our phenotypes, conditions we manifest is encoded in the DNA, we need to know what the DNA is. So without even having the book in front of us, we cannot look for the right page and see what paragraph is right now working on us. So the Human Genome Project was a result of this recognition that we need to know the entire blueprint of human.
So they mapped it.
Exactly. So there are 3 billion letters, ACGT, in the human genome. In every single cell, there are about 40 trillion cells in our body. Every cell has 3 billion letters of DNA. So the goal of that project was to read from left to right those 3 billion letters.
And did you use computers in processing it?
Yes. So it's it takes a combination of biotechnology and computational technologies, because there is no technology which can just go in the cell and start reading ACGT from 1 to 3 billion. So the way it's actually done is things are broken into tiny little pieces. And then what you end up is a jigsaw puzzle which has to be put together to make the whole picture. So the biotechnology gives us the pieces, and the computer technology puts the pieces back together to make the whole picture, which is the book of life.
Very complicated.
Yeah.
So we have one piece of a contribution. What about James Watson and Francis Crick?
Francis Crick and Watson, so they go back to 1953. So in 1869, someone in Switzerland first recognized there is something that does not look like proteins from the bandage of a patient. Under the microscope, they looked at this thing, there is something that's not protein. So that was the first observation of this thing called DNA. It took almost 100 years, 1943, when they recognized this substance as something that can transform cell into another type of cell. So this material had this transforming power. So that was a recognition of what this might be. It could be hereditary material that passes on traits between people. And but still, it was not known, what does this thing look like? What is its structure? So Crick and Watson were the ones who solved the structure of DNA, this double helix, that we now know very well, and that has beautiful properties that puts it all together. It helps it replicate in a very robust way, and that's the basis for replication and life in general.
So what is DNA?
What is DNA? Deoxyribonucleic acid. It's a long chain of 4 types of basic building blocks. Just keep putting them together. But the way they are, the sequence of these letters basically is a sentence in DNA language that tells the cell or the body what to do in a given situation. Like right now, I'm talking to you. So some DNA in my body right now telling my brain to tell my vocal cords to make these sounds. So all this is happening. It's all encoded in the DNA.
It's encoded. The RNA is?
RNA is ribonucleic acid. So the way it works in our bodies, DNA is the blueprint. That DNA has to be converted into proteins that do the actual work, right? So the instruction has to be read from the DNA, and they go into the assembly line factory, and out comes the protein that does the work. So this process from DNA to protein, it has an intermediate stage that's called RNA. So DNA is read, and RNA is produced, one for each protein, and then that is translated into what the protein that works.
So it's the workhorse,
it's a workhorse,
essentially,
yeah,
of the DNA. What is non-coding DNA? If you just talk about that in relationship to what you're doing at the University of Maryland.
Absolutely. So in the 3 billion base pairs, earlier we thought that proteins is all that matters. Okay, so the piece of DNA that actually makes a messenger RNA that makes a protein is all that matters. But if you just look at the whole genome and look at the portion that makes proteins, is 3% of the genome, 2 to 4%, depending on who you talk to. So there's 96% of the genome that is not coding for a protein. So that's what we refer to non-coding DNA. But then there is a whole debate how much of this non-coding DNA is doing anything at all.
Yes.
Right? And we now know very well that a large portion of those non-coding part is actually telling this coding part when to get activated.
Turning on and
Turning
off.
on and off. So all that information about when to use each protein is encoded in the other 96% of the genome.
Tell me the benefit of this study. You had talked about, I mean, actually what got my attention was the press release from your department. It says a new method identifies genetic factures that contribute to heart disease or the risk for heart disease.
Absolutely, yes.
I guess without getting too complicated, I can see the benefit of that if you can identify a gene that is actually causing heart disease, right?
So
or susceptible to heart disease.
Right. So in very general terms, that is the eventual goal of Human Genome Project, is to find all the locations in the genome at which how people differ determines whether they are susceptible to disease or not, whether they get Huntington disease or not, whether they have congenital blindness or not. So the goal is for all genetic diseases to find those locations which not only correlate, but actually determine the disease. So that's the ultimate goal of this entire endeavor, right? Now, the simplest situation will be that there is a protein and that's broken because of a mutation, and protein is not produced or is not produced the right form of the protein, and that causes the disease. A classic example is Huntington's disease. So here there is a mutation, there is a lot of repeats are inserted in the protein, and what the protein you get is very tangly. It's called tangled up, and that's what accumulates in the brain and all that and causes the disease, for example, right? But if you just focus on the changes in the coding part, then you're very limited to small fraction of human diseases. Because a vast majority of what we call complex diseases, like heart disease, diabetes, and schizophrenia, and all these things, lots of genes are involved. And it's not the specific genes are broken, but the instruction that tells this gene to be active or inactive, that instruction book is broken, right? So now we need to shift our attention from protein mutation to regulatory or non-coding genome mutations, right? So that's a whole field now. How do we find non-coding mutations that are correlated or underlie diseases, right?
And I understand you've had a measure of success. Tell us about that. Where you've actually moved quite a distance from the old days of Watson and those guys.
Right. We as in
the whole scientific community.
Yes. Right. We can come later to that how that has become possible.
Sure.
But specifically with regards to our study rather. So the protein is the ultimate, workhorse. The instruction that tells the protein to be made or not to be made is what we are after.
Yes.
So in a way, if you could identify the mutations that change the protein level, divorced from any disease context, okay, that would be a huge benefit, as you can imagine. So for any disease, you can ask, give me all the mutations that really changing the protein levels in this particular context. If it's a neurological disease, we will look at the nerve cells. If it is heart disease, we will look at the human heart. But what we are interested in is all the genetic variations that determine gene activity level differences between people.
There's got to be millions.
There got to be millions, exactly.
Yeah.
So that whole field, it has a name. It's called EQTL, Expression Quantitative Trait Loci. So it's just like, it's very analogous to when you're trying to ask what mutations correlate with heart disease. I'm going to ask a slightly different question. what mutations correlate with gene activity level.
I see.
Not disease, but gene activity level. But if this gene is known to be involved in heart function, then we are getting somewhere already by asking this different question.
Sure, sure.
Right? But the good thing about this different question is we understand at a mechanical level how this process works. Right? So the traditional approach to do that was purely statistical. So you get 1,000 people, get their gene activity level, get their genetic variations.
I think you did something like 2,000, right?
Yeah, we only worked with 2,000 genes, but in general, you would get the gene activity level across all these people and look at all the variations and correlate every variation with every gene activity level across people. But what we did was we said, okay, this purely statistical approach really misses on the fact that we understand the molecular mechanisms, how this process works. So we brought in an entire new layer of our understanding of the mechanism to make this process much more efficient, much more accurate.
How do you do that?
How do you do that? So that will require a micro lecture on linkage in the genome, but I will try, okay?
Brief.
Yes. So the way all our genomes, if you may imagine, come from one place. Back in the time, who knows when, there was one piece of RNA or DNA. That same piece keeps getting replicated and duplicated and mutating, eventually has given rise to all life forms on Earth. Okay? Now, if you can imagine, and things break and things get rejoined and all that, if two things on the genome are nearby, the chance of them separating from each other is small, just by chance, right? So this is the basic principle to identify culprit genes for any disease. So what you're looking for in the disease, people with disease and healthy people, what are the commonalities in terms of genetic variation? So once you identify, here is the locus. Every time there is an A here, this person has a disease. Every time it's a T here, this person does not have a disease. So when you do that, you know that the real culprit gene may not be that thing, but somewhere nearby, because things travel together in the DNA across evolution over time. So that's the basic idea behind identifying disease genes. Okay? But when you do that, you have lots of variation in short distances, then you don't know who is the culprit here. They're all equally culpable. What we do is we ask a slightly more involved question now. So here are the candidates. Now, which one is more likely to actually change the activity of this gene? And to answer that question, we bring in lots of additional knowledge about how specific locations tell the gene to turn on or off, right? And that level of information we call epigenetics. So this is not the genome, it's the epigenome. It's the way the genome is modified that changes its activity. And we know how something is modified when it actually works as a regulator, when it has the power to tell the gene to get turned on or off. We know these things. So we look at that additional layers of information, and then we can say, okay, here are the 10 candidates. These 9, I don't think these are the real ones, because they don't have the characteristic to tell this gene to turn on or off. So it must be this 10th one, because it has all this additional information.
Interesting. Now, so since I've been reading about that you identified a gene variation that actually affects some common blindness, genes that actually affect common blindness. Can you explain that to me?
That's not my study, obviously.
Okay,
that's not my study at all.
It just seems that it's a part of the report.
Right.
So giving us hope.
Yeah, I can tell you something. So that is one of the many successes of this whole endeavor, to be able to identify a specific gene that is clearly underlying a disease. So congenital blindness. So what happens is a particular gene is broken. It has a partner, it's a twin, which compensates for it in the rest of the body.
Yes.
But in retina, that other partner is not present. So when this first gene breaks, then we are in trouble. That leads to congenital blindness. So a simple solution, quote unquote, is to just inject that gene into the retina.
Yeah.
So that has been the
Beneficial.
current approach for therapy. It's called gene therapy. You actually put the good gene in a what we call a vector, viral vector, and inject it in the retina.
What is the team that you lead? What is it called?
The team that led that work?
Yeah, no, the team that you are working with at the university.
At Maryland or at the University of Pennsylvania. I have my own team.
I see.
but this the hard work is in collaboration with a large group of researchers at University of Pennsylvania. Okay. It's something called a magnet consortium, and the goal of this consortium was to gather hundreds and hundreds of human hearts, failing hearts that had to be taken out of the body, and healthy hearts that were donated but could not be used for various reasons. So we have human hearts, healthy hearts, and failing hearts, and all this research that we published is using that data. So that's why the variations we identify that regulate the activity of genes in human heart have real implication in using them to study heart failure, actually.
And you said it was a publication produced. Where can one read it?
The publication, this is in Nature Communications.
I see, yes. This is so fascinating and it's so interesting, but I almost, as a communicator, it's hard for me to get my entire understanding of it. But I'm beginning to understand it even more. Thank you for coming to share your data with us. The Genome-wide Association study, what is that?
So that is precisely this idea of finding the genetic variations that are associated with a phenotype. Any phenotype, height, color, fur coat of dogs, what have you. So as the name suggests, it's genome-wide and it's association. So you take any trait of your interest. So let's just for simplicity, heart failure. So you recruit heart patients and you recruit healthy people who are matched for various gender and age and such. You get their blood, you get their genotype, which is a short form of genome, basically. Okay. So you have, let's say, 1 million locations in the genome where people differ from each other. So for each of these 1 million locations, you compare the healthy cohort with the heart failure cohort. And you ask whether at that one location, they have different piece of DNA. And you do that for each of those 1 million locations. And the locations that come up highly significantly different between these two groups, those are your gateway to further exploration to finding the cause of the disease.
What are the tools used for this research?
What are the tools used? In a sense, it's very simple. The hardest part in this whole endeavor is recruiting people.
Yes.
That is the hardest part. That's what takes most of the money. Once you have the people, then the basic genome-wide association study just requires any source for DNA. It could be cheek swab, it could be blood. It doesn't have to be invasive at all. All you need to know is the genome, not the genome, but the genotype of this person, and that can be done very easily. It still takes money, but there is no technical challenge there per se. And once you have that, and you have done the job well in terms of grouping the people in the right groups, then it's a very simple statistic by looking at each of those locations and seeing which location comes out significantly different between these two groups. But the hard part starts after that.
Once you have the data.
Yeah, so the GWAS itself is just this part that I just mentioned. It's just getting to the candidate loci in the genome, locations in the genome, which may be underlying the disease, right?
What excites you most about this field?
I'm just a kid in a toy store, basically. That's what it boils down to. There are so many unanswered questions. I'm mainly turned on by basic question: how does it work? Who has done it? That kind of question. So, as I mentioned in my presentation from Maryland, we have a complex animal, and we have hundreds of different cell types. My eye, my skin, my teeth, my heart. Each one does a very specific thing, and they all start with a single cell.
A single cell. Yes.
After conception,
Yes.
and that single cell somehow develops into this complex organism doing so many different things. Is the same blueprint in every cell, but each cell does something very specific. So who is controlling all this process? We know that there is instruction book for the genes to be activated. Who is instructing the instruction book? Right, it has to start somewhere. So there is a built-in, robust feedback mechanism that takes the single cell into these different paths, making the full animal.
This is fascinating. Is there, this is just so incredible that I wish I had another scientist to also walk us through it. But tell me about just a general question, the scientific method. Why is it important? Just in a general sense, why the scientific method is important?
Scientific method as in general, or
yes.
my particular field?
As it relates to your field.
Well, scientific method is the
mother of all of it.
Mother of all of it. The best way we know how to interrogate. Once you have a question, we have to have a system to get to the answer. And the scientific method, all it says is the way to get there without giving you any specifics. But as it applies to my specific field, you apply the same general principles to, for example, if we claim that we have found a genetic variation that changes the activity of this gene, then the scientific method would say that go in the lab and make this change in a piece of DNA, see if that changes this.
Yes.
We cannot do that with humans, obviously, but we can do experiments like this in mouse to really establish that what I'm finding is true. So that's all scientific methods tells me.
That's exactly right.
Yeah.
So tell me, I usually ask our scholars near the end of the program, what is a book that a student might read that would further their knowledge, a basic book that would further their knowledge on genetic, the history of DNA and history of
Things
genetics?
we're talking about.
Right.
I think the best resource these days, honestly, for an outsider, for a layperson, Wikipedia is fantastic. It's fantastic.
Yes.
And NIH, National Institute of Health, on their website, they have lots of educational, basic resource and information about all these kinds of issues I'm talking about. So that's a fantastic resource. That's the reason it exists, to educate the public about genetic research.
About what's going on.
Yeah, the genome and the genetics and the diseases.
Dr. Hanan, I have to say it properly. Say it for me.
Hanenhalli.
Hanenhalli. Dr. Hanenhalli is in fact a professor at the University of Maryland. We've been talking about the new discoveries of genetics, and you'll be able to learn more about the Scholars Chair by going to Read One YouTube at online, and you can also follow us on Twitter and Facebook. I am Khalil Shadid. Good night.
Hit it, hit it, hit it. Amazing.